Welcome to this deep dive, the RadBio edition of Rad Onc Smart Review.
Great to be here.
Today, we're going to tackle a really critical area for, well, everyone in Rad Onc. That's the tumor microenvironment.
Right.
We'll be focusing specifically on tumor hypoxia, those tricky low oxygen zones inside a tumor, and also the process of reoxygenation that happens during radiation therapy.
Absolutely. Understanding these is it's fundamental. Hypoxia is such a huge piece of the puzzle, often a frustrating one when it comes to treatment effectiveness.
Definitely. So let's jump right in. What exactly is tumor hypoxia? And, what's the main reason tumors develop these oxygen-starved areas?
Okay, so tumor hypoxia, in simple terms, just means parts of a tumor have way less oxygen than normal, healthy tissues.
And why does that happen?
The main driver is really the tumor's own growth. It grows fast, often chaotically.
Too fast for its own good?
Exactly. The cancer cells are dividing rapidly. They need loads of oxygen. But the tumor's ability to grow new blood vessels, that process we call angiogenesis, just can't keep pace.
So it tries to build a supply network, but it's not good enough.
Precisely. And the network it does build is often, well, structurally flawed. Leaky, disorganized vessels. So you get areas with poor blood flow and bam, hypoxia.
Makes sense. So it's like the tumor's growth outpaces its plumbing.
That's a great way to put it. And there's a physical limit, too. Oxygen from the blood can only diffuse so far into the tissue before the cells gobble it up.
Right. There's a limit to how far it can travel. What's that distance, generally?
In tumor tissue, the number you need to remember is about 150 micrometers. That's the maximum diffusion distance from the arterial end of a capillary.
150 micrometers? I feel like I've seen a different number sometimes, like 70.
Yeah, you might see 70 micrometers mentioned, especially older texts. But the current evidence really points more towards 150 micrometers as the limit from the arterial side. It's obviously a bit less further down towards the venous end.
Okay, 150 micrometers. That sounds incredibly small when you think about the size of a tumor.
It is. And this limited diffusion range, it gives rise to a model we use to understand this, the tumor cord. Heard of that?
Yes, the tumor cord model. Can you walk us through that?
Sure. So imagine a single small blood vessel running through the tumor tissue.
Okay.
Around that vessel, you get these sort of concentric zones of cells based on their oxygen level.
Starting right next to the vessel.
Yep. Right up against the vessel wall, you have the normoxic zone. These cells are getting plenty of oxygen. They're usually dividing actively, and importantly for us, they're quite radiosensitive. Radiation kills them relatively easily.
Okay, makes sense. What happens as you move further away?
As you move out, beyond that, say, 150 micrometer diffusion limit, you hit the hypoxic zone.
The problem area.
Exactly. Here, oxygen levels drop way down. The cells are still alive, they're viable, but they're starved for oxygen. They might be quiescent, not dividing much, or cycling very slowly. And here's the kicker. These hypoxic cells are radioresistant, much harder to kill with our standard radiation.
Right, that's the core challenge. And is there anything beyond that zone?
If you go even further from the vessel, where oxygen just can't reach at all, you get the anoxic or necrotic zone.
So completely depleted.
Totally depleted. These cells are dead or dying necrotic. Now, we're not directly targeting these dead cells with radiation, but they're still part of the microenvironment, contributing to pressure and other factors.
So that hypoxic zone, viable but radioresistant, maybe just a few cell layers thick, sometimes that's the real hurdle for RT.
That's it in a nutshell.
Okay, so that paints a clear picture of this sort of stable, diffusion-limited hypoxia. But isn't there another type of hypoxia too, something more transient?
You're absolutely right. We need to differentiate between chronic hypoxia and acute hypoxia.
Okay.
Chronic hypoxia, sometimes called diffusion-limited chronic hypoxia, is exactly what we just described. It's stable, persistent low oxygen, simply because the cells are too far from a working blood vessel. If things don't change, these cells eventually die and become necrotic.
Got it. So what's acute hypoxia then?
Acute hypoxia, or perfusion-limited acute hypoxia, is quite different. It happens because of temporary interruptions in blood flow within those abnormal tumor vessels we mentioned.
The faulty plumbing acting up again.
Exactly. These vessels can just, temporarily collapse or get blocked, or the flow just becomes erratic. So a region of the tumor suddenly loses its oxygen supply. That's acute hypoxia.
But it's temporary.
Often, yes. If the vessel opens back up, or the blockage clears, oxygen levels can shoot back up to normal, or near normal. So cells in these areas can cycle back and forth between low oxygen and, well, better oxygenation. Think of it like a flickering light bulb for oxygen supply.
Okay, like intermittent kinks in the pipes. And I guess both types, the chronic steady kind and this acute flickering kind, both contribute to the overall radioresistance problem.
They absolutely do. Both make the tumor harder to treat effectively with radiation.
So given how important this is, how do we actually, see or measure hypoxia in a tumor? Can we detect it?
That's a critical question.
Yeah.
And yes, we have several ways to try and detect and quantify hypoxia.
Like what? Can we stick a probe in?
Well, yeah, actually. One direct method uses oxygen probes. These are tiny electrodes. The Eppendorf probe is a common example that you can literally insert into a tumor.
Wow.
And they directly measure the partial pressure of oxygen, the PO2, right at that spot. Gives you a very localized real-time reading. The Eppendorf is known for being pretty fast responding.
But I imagine that's quite invasive.
Extremely invasive. So it's mostly used in research, maybe
Yeah.
sometimes in very accessible human tumors, but it's not routine clinical practice.
Okay. What about things we can use more clinically? I've heard about labeled markers.
Right. The labeled nitroimidazoles. These are probably more relevant day-to-day, or at least in clinical trials and advanced imaging.
How do those work? Misonidazole, promonidazole, those kinds of things.
Exactly. So compounds like misonidazole, that's M-iso-NID-uzole, monidazole, pimol-NID-uzole, and fazafluoroisomycin are rabinoside. They start off non-toxic.
Okay.
The clever part is inside cells that are low on oxygen, specific enzymes chemically reduce a part of the molecule, the nitro group.
Only in hypoxic cells.
Primarily, yes. Under normal oxygen, this reduction doesn't happen efficiently. But in hypoxia, it gets reduced, and this reduced form is reactive. It then sticks, binds covalently to proteins and other big molecules inside that hypoxic cell.
So it gets trapped there.
It gets trapped specifically in the hypoxic cell.
Clever. And then how do we see where it's trapped?
We attach labels to them. For imaging, we can use radioactive isotopes like fluorine-18 for FISO or FASA, which lets us use P scans.
Positron emission tomography.
Right, or iodine-123 for IAZA for SPECT imaging. These scans can then show us maps of where the hypoxic regions are within the tumor, non-invasively.
That sounds very useful. What about in tissue samples?
Yeah, we can also use fluorescent tags. Pomonidazole is often used this way. You treat the animal, or sometimes use it in patient biopsy studies, take a tissue section, stain for the bound pomonidazole, and you can see it under a microscope.
What does that look like?
Typically, you see this like green rim of staining. It shows up right where you'd expect that hypoxic zone to be in the tumor cord model, sandwiched between the well-oxygenated cells closer to the vessel, which don't stain, and the necrotic core further out. It's a really neat visual.
That really confirms the model, doesn't it? What about HIF-1 alpha? That comes up a lot with hypoxia, too.
Ah, yes. HIF-1 alpha, hypoxia-inducible factor 1 alpha. Very important. It's a transcription factor.
Meaning it turns genes on or off.
Exactly. Under normal oxygen conditions, HIF-1 alpha gets made, but it's broken down really quickly. It doesn't hang around.
Okay.
But when a cell becomes hypoxic, HIF-1 alpha becomes stabilized.
Yeah.
It doesn't get degraded. It builds up, goes into the nucleus, and switches on a whole suite of genes that help the cell adapt to low oxygen.
Like genes involved in making new blood vessels. Angiogenesis.
Precisely. Angiogenesis genes, genes for switching metabolism, lots of survival stuff. So if you stain a tissue section for HIF-1 alpha protein, often using a red stain, it tells you that those cells are, or have recently been, hypoxic.
And where do you usually see that staining?
You often find the red HIF-1 alpha staining in or very near those green pomonidazole positive hypoxic areas we just talked about. They overlap quite a bit, confirming the hypoxic status.
So using these tools probes, nitroimidazoles, for imaging or staining, HIF-1 alpha staining, we can get a handle on how much hypoxia there is.
Right, and that leads to the idea of the hypoxic fraction.
Which is just the percentage of hypoxic cells.
Essentially, yes.
Hmm.
The proportion of cells in the tumor that are hypoxic at any given moment.
Yeah.
But the really critical thing to understand about the hypoxic fraction is its variability.
How variable.
Hugely variable. It differs massively between different cancer types. It differs between tumors of the same type in different patients. It even varies within different parts of the same tumor.
Wow. So it could be tiny or it could be huge.
Exactly. Some tumors might have only a few percent hypoxic cells. Others might be 50% or even more. This heterogeneity is a massive challenge for predicting radiation response.
Okay, so we've established hypoxia is common, variable, and makes cells radioresistant. Sounds pretty bleak. But there's a silver lining, right? Reoxygenation.
Ah, yes. The cavalry, in a sense.
Mm-hmm.
Reoxygenation. It's one of the famous 4 R's of radiobiology.
The 4 Rs, let's list them quickly. Repair,
Repair of sublethal damage, repopulation,
repopulation of cells between fractions, redistribution.
or reassortment, meaning cells moving into more sensitive phases of the cell cycle. And the fourth R,
Reoxygenation, the one we're focusing on now.
that's the one. And it's incredibly important for why fractionated radiotherapy works.
Okay, break it down for us. How does reoxygenation actually happen, step by step?
All right, so imagine we deliver a dose, a fraction of radiation, to the tumor.
Zap.
Now, as we said, the well-oxygenated normoxic cells are the most sensitive. So that first dose preferentially kills them off.
Okay, takes out the easy targets first.
Right, which means if you were to look immediately after that dose, the proportion of hypoxic cells among the survivors have actually gone up.
Counterintuitive maybe, but makes sense. You killed the oxic ones.
Exactly. So, right after the zap, the tumor might look even more hypoxic overall, fractionally speaking.
But that's not the end of the story.
Not at all. Over the next few hours, the time frame is typically within about 6 hours or so, things start to change.
What happens?
Well, first, the cells that were killed start to get cleared away. The body's cleanup crew gets to work. The tumor might shrink a tiny bit. And crucially, with fewer living, respiring cells around.
The overall oxygen demand drops.
Precisely. Less demand for oxygen. So the oxygen that is still being delivered by those remaining blood vessels can now diffuse further. It can reach cells that were previously just beyond that 150 micrometer limit.
So the surviving hypoxic cells suddenly find themselves with more oxygen available.
Exactly, they reoxygenate. And what happens when a hypoxic cell gets more oxygen?
It becomes more radiosensitive again.
Bingo. Becomes more vulnerable to the next fraction of radiation we deliver.
So it's a dynamic process. Kill oxic cells, oxygen demand drops. Hypoxic cells get oxygen, they become sensitive, then the next dose can kill them.
That's the cycle. And it can repeat, ideally, with each fraction of a standard radiotherapy course.
So fractionation isn't just about letting normal tissues repair, it's actively leveraging this reoxygenation in the tumor.
That's a huge part of it. Experiments have shown this quite elegantly.
Like in animal models.
Yeah, classic studies use things like transplantable mouse sarcomas. They give a hefty dose, say 10 gray. Right after, the anoxic fraction might shoot up to nearly 100%.
Oh.
But then, wait about 6 hours, measure again, and that anoxic fraction could plummet back down, maybe close to the original baseline level, say around 18 or 20% in that particular model.
So significant reoxygenation happening within hours.
Relatively quickly, yes. It's interesting, the peak for reoxygenation seems to be around that 6-hour mark, which is generally a bit slower than the peak for sublethal damage repair, which is more like 2 to 4 hours.
That timing difference could be important for scheduling, maybe, like with hyperfractionation.
Potentially, yeah. It factors into the thinking behind different fractionation schedules. But, and this is a big but, we have to remember, reoxygenation isn't guaranteed, and it's not the same everywhere.
How so?
The extent and the speed vary massively. Different tumor types, even in mice, behave differently. Some reoxygenate really well and fast. Others might reoxygenate fast, but not completely. The hypoxic fraction drops, but not back to baseline.
And some barely reoxygenate at all.
Exactly. Some tumors, classic examples are osteosarcomas in mouse models, are notoriously poor reoxygenators. They reoxygenate very slowly, if at all, and that hypoxic fraction stays stubbornly high.
Which likely explains why some tumor types are just harder to treat with radiation.
It's definitely a major contributing factor. This variability in reoxygenation is key.
Okay, so understanding this interplay, the problem of hypoxia and the potential solution of reoxygenation really impacts how we think about treatment, doesn't it?
Absolutely. I mean, the very existence of hypoxia is why many solid tumors resist standard radiation, low LET radiation like X-rays.
Right. And the fact that reoxygenation can happen is a cornerstone of why we use fractionated radiotherapy.
Totally. Splitting the dose into smaller bits over time, usually daily, gives those hypoxic cells that chance to get oxygenated and become sensitive for the next hit. Even shorter gaps, like 6 to 8 hours in hyperfractionated or accelerated schedules, can allow some reoxygenation.
And this benefit is mainly for the tumor, right? Normal tissues are mostly well oxygenated anyway.
Generally, yes.
Yeah.
Most normal tissues don't have a significant chronic hypoxic problem, so the main benefit of allowing time for reoxygenation is thought to be enhanced tumor cell kill.
Okay, but what if we have a tumor where we suspect it's really hypoxic and maybe doesn't reoxygenate well, like those osteosarcomas you mentioned? Are there specific strategies then?
Yeah, that's where hypoxia-modifying strategies come into play, things people have tried or are researching. Well, there are hypoxic cell radiosensitizers. These are drugs designed to specifically make hypoxic cells more sensitive to radiation, sort of mimicking oxygen.
Okay.
Then there are hypoxic cytotoxins, drugs that are actually more toxic to cells when oxygen is low. They preferentially kill the hypoxic cells.
Interesting. What else?
You could think about using different types of radiation, high LEET radiation like neutrons or carbon ions,
Why does that help?
because their cell-killing ability is less dependent on oxygen. They have a lower oxygen enhancement ratio, or OER. So the presence or absence of oxygen makes less difference to how effective they are.
Got it. Any other approaches?
And then there's the idea, especially with better imaging, of dose painting.
Dose painting.
Meaning if you can reliably image the hypoxic parts of a tumor, say, with that F-Einzo PET scan we talked about, maybe you could deliver a higher radiation dose specifically to those resistant areas while keeping the dose lower to the better oxygenated parts.
Targeting the tough spots.
That's the concept. Still largely investigational, but a very active area of research.
It's also worth mentioning, isn't it, that sometimes a simple blood test can give a clue about hypoxia, like hemoglobin levels.
That's a really valuable clinical point, yes.
Yeah.
Lower pretreatment hemoglobin levels in patients' anemia have been linked to worse local control after radiation in several cancers: head and neck, cervix, lung.
And the thinking is, lower hemoglobin means less oxygen carrying capacity in the blood overall.
Which could lead to worse tumor hypoxia, making the tumor more resistant. It's an indirect indicator, but clinically relevant.
So pulling it together, reoxygenation is great when it happens, and we rely on it for standard fractionation. But if it doesn't happen well, or the hypoxia is too severe, we need to think about other strategies.
Precisely. Standard RT implicitly banks on reoxygenation working reasonably well. Advanced techniques like dose painting might offer ways to tackle tumors where it doesn't.
Okay, fantastic overview. Let's transition now to some clinical pearls, those key nuggets for residents to really lock in.
Sound good. Pearl number 1.
Clinical Pearl 1. Tumor hypoxia is a major cause of radioresistance, especially to our standard low LET radiation, like X-rays. Remember that resistance factor?
Absolutely. Clinical Pearl 2.
Clinical Pearl 2. Remember the distance. Oxygen can only diffuse about 150 micrometers from a capillary into respiring tumor tissue. That limited diffusion is why chronic hypoxia develops.
150 micrometers. Got it.
Oh.
And the third pearl.
Clinical Pearl 3. Reoxygenation is your friend. It's the improvement in oxygen for hypoxic cells between fractions, and it's a key reason why fractionation works so well for many tumors. It helps overcome that initial resistance.
Excellent pearls. Hypoxia resistance, 150 micrometers equals limit. Reoxygenation, fractionation benefit.
Perfect. Now ready for a quick board blitz? Test ourselves.
Let's do it. Fire away.
Okay, question 1. Which of the following best describes chronic hypoxia in a tumor? A. Temporary lack of oxygen due to intermittent blood vessel closure. B. Increased oxygen levels due to radiation-induced cell kill.
Well,
C. A stable state of low oxygen due to cells being located beyond the oxygen diffusion limit from blood vessels. D. Oxygen levels found in rapidly proliferating cells immediately adjacent to capillaries.
Okay, chronic hypoxia. That's the stable kind due to distance. So I'm going with C.
And why C?
Because chronic hypoxia is diffusion limited, based on how far oxygen can travel from the vessel. Option A is describing acute perfusion-limited hypoxia. B is sort of the result of reoxygenation after cell kill, and D is describing the normoxic zone, so C it is.
Correct. Question 2. What is the approximate maximum distance oxygen can typically diffuse from the arterial end of a capillary into respiring tumor tissue before being consumed? A. 10 micrometers. B. 50 micrometers. C. 70 micrometers. D. 150 micrometers.
Okay, we hammered this one. It's the more updated accepted value. D, 150 micrometers.
Rational.
While 70 micrometers pops up in older stuff, 150 is the current figure based on evidence for the arterial end diffusion limit in respiring tumor tissue.
Spot on. D is correct. Question 3. The process by which previously radioresistant hypoxic tumor cells become more radiosensitive between fractions of radiotherapy is known as A. Repopulation.
Yeah.
B. Repair of sublethal damage. C. Reassortment. D. Reoxygenation.
Ah, one of the 4 R's, the one that improves oxygen status and sensitivity. That has to be D, reoxygenation.
Explain why it's D.
Because reoxygenation is specifically defined as the improvement in oxygen levels in hypoxic cells between fractions, which increases their sensitivity to the next dose. The others are distinct processes. Repopulation is cell growth, repair is fixing DNA damage, reassortment is cells moving through the cell cycle.
Excellent. D is correct. Final question number 4. A researcher wants to identify hypoxic regions within a tumor biopsy using a chemical marker that selectively binds to cells under low oxygen conditions. Which of the following agents would be suitable for this purpose? A. Propidium iodide. B. BRDU. C. Hematoxylin. D. Pomonidazole.
Okay, looking for a hypoxia marker that binds. We talked about nitramidazole doing that. Pomonidazole was one of them, so I'll go with D-pomonidazole.
And why pomonidazole?
Because it's a nitramidazole that gets reduced specifically in low oxygen, becomes reactive, and binds there, allowing us to detect those hypoxic cells. Propidium iodide stains DNA, usually in dead cells. BRDU labels dividing cells. Hematoxylin is just a general nuclear stain. None of those are specific for hypoxia.
Perfect reasoning. D. Pomonidazole is correct. Well done on the board blitz.
Good questions. They really tie the concepts together.
So we've covered a lot of ground, the causes of hypoxia, the types, how we detect it, and this crucial process of reoxygenation. Thinking bigger picture, what are some of the ongoing challenges or controversies here? Where is the field moving?
Well, a major ongoing challenge is still reliably imaging hypoxia in patients, routinely, across all different tumor types.
Right, you mentioned FISO PET, but it's not everywhere, is it?
Exactly. It's not universally available. The spatial resolution can be a limitation, and just standardizing the quantification is still being worked on. Plus, hypoxia is dynamic. It can change during treatment, which makes targeting it even harder.
A moving target.
Pretty much. And in terms of advances, there's obviously huge interest in developing better drugs.
Newer radiosensitizers or cytotoxins?
Yeah, next-generation hypoxic cell radiosensitizers that are maybe more effective or less toxic, novel cytotoxins. And also really refining how we integrate hypoxia imaging into treatment planning. Can we make dose painting a clinical reality that actually improves outcomes? That's a big question.
Lots of work still to be done then.
Definitely. But our understanding is getting deeper all the time, which
Yeah.
hopefully leads to better treatments down the line.
Okay, so let's try and recap the main points from our deep dive today.
Sounds good.
All right, so first, tumor hypoxia. It's common, caused by tumors outgrowing their blood supply, and that fundamental oxygen diffusion limit, remember, about 150 micrometers.
Right. And it comes in two main flavors: chronic diffusion-limited hypoxia and that fluctuating acute perfusion-limited kind. Both lead to radioresistance.
Big problem for standard radiation, but the key counter process is reoxygenation.
The crucial R-killing oxic cells reduces oxygen demand, allowing oxygen to reach deeper, resensitizing those previously hypoxic cells between fractions.
And that's a major reason why fractionated radiotherapy works, though we need to remember that reoxygenation varies a lot between tumors.
Absolutely. And we have tools to detect hypoxia probes, nitramidazole tracers for imaging like BAMISO PET, or staining like PEMO, and HIF-1 alpha staining, which are increasingly important for understanding tumors and maybe guiding treatment, perhaps with things like dose painting or specific hypoxia-targeting drugs if reoxygenation is poor.
Excellent summary. Really covers the core principles that are so vital for us in radiation oncology.
Couldn't agree more. Foundational stuff.
Indeed. All right, that wraps up this deep dive.
Don't forget, you can complete practice oral boards over at radoncsmartlearn.com. And definitely subscribe to Rad Onc Smart Review for our next session. Thanks for tuning in.